Hello everyone, this video is about excretory system of insect. Excretory system in insects is morphologically intimately associated with the organs of digestion. It is well adapted for elimination of various excess and toxic, mostly nitrogenous waste products of metabolism, and for efficient maintenance of constant level of salts, water and osmotic pressure of the hemolymph. The excretory system is basically composed of two structures, the Malpighian tubules and rectum. The Malpighian tubules absorb the nitrogenous waste, water, and salts from the hemolymph, while the rectum performs selective reabsorption and the end product of excretion, the urine, is discharged out. So, first, we are going to learn different types of excretory systems. Anatomical association. The Malpighian tubules are long, slender, convoluted tubes of which the proximal end opens at the junction of hind and midgut, while the distal end remains blind. On the basis of association or dissociation of distal end of the Malpighian tubules with the rectum, two types of excretory systems can be recognized. Basic nephridial systems. In a large number of insects, the distal ends of the Malpighian tubules bathe freely on the hemolymph of hemocoel. The Malpighian tubules absorb various waste substances, salts, and water from the hemolymph and discharge into the hindgut through their distal end. The superior or anterior tubules in Dacus and Drosophila bear distinct dilation at their distal ends. All tubules often open independently, but in some insects such as grillets, all tubules open proximally into a common ureter and the latter finally opens into the gut. Similarly, in some beetles, Haltica and Donartia, a group of 4 tubules open into a common ampulla, while remaining 2 open independently. In the case of red cotton bug, all tubules of one side open into a common ampulla or bladder, which ultimately opens into the gut. In Hemiptera, tubules are differentiated into the distal clear part filled with fluid and proximal opaque half containing solid or granular substance. So first, basic nephridial system we mentioned, then cryptonephridial system. In some insects, the distal end of the Malpighian tubule is closely associated or inserted into the rectum, forming the cryptonephridial or cryptosolanor system. It is formed in the larvae and adults of Coleoptera, larvae of Lepidoptera, some Hymenoptera and Neuroptera. In Coleoptera, the Malpighian tubules do not differentiate into the regions, but in Lepidoptera, the tubules are typically differentiated into two sections, distal part with fluid and proximal part with solid particles. In Coleoptera, the distal ends of the tubules are folded and closely applied to the rectum, and both are enclosed together by the perinephric membrane. The perinephric membrane encloses the perinephric space between the tubules and the rectal wall. The tubules dilate in the perinephric space and the tubules wall thus become very thin and composed of leptophragma cell. In Lepidopteran larvae, the inner layer of tubules is closely applied to the rectal epithelium. The distal ends of Malpighian tubules are folded back in such a way that they constitute an outer layer. It is separated from the inner layer by a 2-cell thick layer of flattened cells. The entire complex is enclosed by an outer membrane and the muscle coat. So that's all about types of excretory system. Two types. First, a basic nephridial system and the cryptonephridial system. And the organs of excretion. The Malpighian tubules. Presence of excretory tubules at the junction of mid and hindgut were first reported by Italian scientist Marcello Malpighi in 1669, and these structures were named later on as a Malpighian tubule by Mackell in 1820. The Malpighian tubules are present in almost all insects except Columbola, some Thysanura, and aphids. Their number, size, and modifications vary among the insects. The basic number of Malpighian tubules is considered as 6. but may undergo reduction or addition in different insects from 2 to 250 and often exist in a multiple of 2. There are 4 in Anoplura, Thysanoptera, Hemiptera, Diptera, 4 to 6 in Thysanura, 6 in Lepidoptera, 8 to 20 in Dermaptera, 250 or more in Dictyoptera, Odonata and Orthoptera, 6 to 20 in ants, 200 or more in Hymenoptera, 2 in Coccidia, but exceptionally 5 in Culicidae. In some groups of insects, Protera, Diptera and Strepsiptera, the malpighian tubules are developed in the form of papillae rather than the tubules. The malpighian tubules are derived from either proctoderm or mesenteron or from an indifferential zone of cells lying between the mid and hindgut. Their development is still a much controversial subject. The wall of malpighian tubule is composed of 4 to 6 large epithelial cells arranged in a circle. The nuclei are distinct and may be palmate or giant endopolyploid of nature. Each cell bears towards the lumen a brush or honeycomb border. Externally, the cells rest on a basement membrane. The basement membrane in some insects like Orthoptera is wrapped externally by spiral strands of muscle. The tubules of Lepidoptera, Diptera and Hemiptera do not possess muscles except at proximal end where a group of circular or longitudinal muscle fibers are loaded. In Coleoptera and Neuroptera, a muscle sheath is continuously running the entire length of tubules, but lacking in Thysanura, Dermaptera and Thysanoptera. There are 2 types of cells in these insects where each malpighian tubule is differentiated into distal clear and proximal opaque regions. The distal part is composed of such cells that bear honeycomb border formed due to modification of the free margins of the cells into 3 to 10 micron long cytoplasmic filaments packed very close together. The proximal part, on the other hand, is composed of those cells which have a brush border formed due to cytoplasmic filaments of 7 to 14 length separated from each other. The plasma membrane of basal region of the cells is deeply invaginated within the cells and much complex in honeycomb border. Mitochondria are conspicuous in filaments of honeycomb border while they occur in large number within the basal folds of plasma membrane and the brush border. The malpighian tubules contain some enzymes such as alkaline phosphatase, acid phosphatase, lipase and succinic dehydrogenase, and also some of the water-soluble vitamins like riboflavin, thiamine, niacin, pantothenic acid and ascorbic acid. In rectum, the epithelial cells from rectal pads or papillae, cryptonephric complex or chlorine epithelium. The rectal pads. Swaminathan first discovered the rectal pads in the rectum, and Wigglesworth found resorption of water from the rectal condensed by these structures. Muscle layer is there, hemolymph will be passing through the above layer, sinus, septate desmosome, identification of basal plasma membrane, intercellular channel, intercellular space, rectal pad cell, septate desmosome and cuticle. In primitive insects, the rectal wall is composed of a single layer of flat epithelial cells or columbola. In Hemiptera, the epithelial cells become thickened as a small wall differentiates into a pad comprising of columnar cells. In Dictyoptera and Orthoptera, the rectal epithelium is regularly differentiated into symmetrically arranged 6 rectal pads. They are discontinued from each other by undifferentiated thin epithelium. Each pad is composed of a single layer of columnar epithelial cells, but in iron membrane members, some secondary cells are lodged in the basal surface of the pads. In Hymenoptera and some Lepidoptera, the rectum bears a large number of small and spherical epithelial pads. Each pad is composed of 2 layers, one of columnar epithelial cells and the other smaller epithelial cells. Both the layers may or may not be separated from each other. Total number of pads may exceed over even 500 within a single insect. In adult Diptera, the epithelial pads are replaced by epithelial cones or papillae. The rectal pads of the cockroach, Periplaneta americana, are studied in detail by some scientists. So each pad is composed of an internal layer of cuticle, middle layer of flattened columnar epithelial cells, and outer layer of muscles. The muscle and epithelial layers are separated, forming a narrow subepithelial sinus. The epithelial cells are themselves separated from each other and give rise to narrow intercellular channels of about 200 angstrom diameter. The intracellular channels are separated from the rectal lumen as well as from the subepithelial sinus by desmosomal connections or septa. The intercellular channels often communicate with large intercellular spaces of 10 to 13 mm diameter. The intercellular spaces are formed due to distal dilations of plasma membrane enfoldings. The tracheal penetrate into the entire width of the rectal pad and they extend the fine tracheoles into the intercellular spaces. The columnar epithelial cells contain large number of mitochondria, mostly aggregated in the lateral and apical plasma membrane enfoldings facing the cuticle layer. The rectal papillae of the adult blowfly Calliphora erythrocephala has been extensively studied by Gupta. Each papillae is composed of a cortex and medulla. The cortical region is formed from a single layer of columnar epithelial cells, while the medullary region is composed of connective tissue. The papilla is externally covered with a thin layer of cuticle facing the lumen. The epithelial cells from the intercellular spaces and sinus form the intercellular spaces and sinus. Both form a continuous channel system which opens into the infundibular space. The infundibular spaces occur in between the cortical and medullary regions. The infundibular space opens into the hemocoel through the semilunar valves, but prevents the backward flow of hemolymph. The papillae are studied with the fine nerves, neurosecretory cells, and tracheae. The tracheoles penetrate into the intercellular spaces. Then the cryptonephric complex. The terminal ends of the Malpighian tubules are enclosed within a perinephric space. The perinephric space lays between an outer double-layered perinephric membrane and an inner rectal peritoneal membrane. The inner perinephric layer is interrupted in a group of specialized cells, the leptophragmata. The leptophragmata constitute a thin wall between the hemocoel and the interior of enclosed ends of Malpighian tubule. Their position can be recognized from the external bulgings found along tubules, the bursofleur. The rectal wall bears two types of spaces, each 6 in number. The outer spaces are lying in between the cryptonephric complex and rectal circular muscle layers and are known as perirectal spaces. Similarly, the inner spaces are occurring in between the rectal circular muscle layer and the rectal epithelium and are known as the subepithelial spaces. The perirectal spaces are separated from one another by the longitudinal muscle bands. The cryptonephric complexes are also separated from each other by the intertubular cells. This arrangement accelerates water conservation by adding absorptive capacity of tubules to that of rectal epithelium. The tubules, on the other hand, collect the ions from hemolymph. The rectal chloride epithelia. In odonate larvae, the basal pads of gill lamellae are modified into the excretory or osmoregulatory epithelia and are commonly referred to as the rectal chloride epithelia. The structures absorb sodium and chloride ions from exogenous water entering through the anus and transport to the hemolymph. In order to maintain osmotic hyperregulation, the structure and function of extensively reviewed by Komnick in 1977. Next, the accessory organs of excretion. The accessory organs of excretion, nephrocytes. They form two major groups, a dorsal group of pericardial nephrocytes and a ventral group of infraintestinal nephrocytes. The dorsal nephrocytes are arranged into a long chain on either lateral side of the heart in the pericardial sinus. This group occur in most of the insects. The ventral nephrocytes form a pair of chains which run along the ventral surface of intestine and finally are attached to the salivary glands. The ventral nephrocytes occur only in the dipteran larva. Some cells are enclosed singly or in cluster in basement membrane and are intermingled in the connective tissue strands which are suspended in the hemocoel. The cells have folded plasma membrane at one end, forming the invaginations like the food processes. They absorb non-particulate colloids from the hemolymph by endocytosis and release various substances by exocytosis. They therefore differ from the hemocytes, as the latter are always necked and move freely in the hemolymph. The nephrocytes are penocytotic, while the hemocytes are phagocytic. They are analogous to the vertebrate reticuloendothelial system. They appear as podocytes and can be recognized in vivo by the administration of trephine blue dye. They are arranged in as garland around esophagus, proventriculus junction ventrally, along heart and aorta dorsally, and are also scattered in the fat body cells and are called as the garland cells, pericardial cells, or disseminated nephrocytes, respectively. They are large cells ranging from 140 to 200 μm in diameter and contain polytene chromosomes. They are derived embryologically from the bilateral coelomic mesoderm of each metameric segment. Ultrastructure of nephrocytes reveal formation of network of the labyrinthine channels by extensive invaginations of the plasma membrane and perform ultrafiltration. Cytoplasm consists of fine tubular elements of 60 to 75 nm diameter, besides the macrotubules of 24 μm diameter and microfilaments of 8 μm diameter. They are equipped with Golgi endoplasmic reticulum, lysosome complexes in the cytoplasm, besides numerous mitochondria and vacuoles of variable sizes. The cytoplasm is rich in glycogen, proteins, and lipid contents. The insect nephrocytes perform renal function analogous to glomerulus, ultrafiltration, and the proximal tubules, endocytosis of the vertebrate kidney. Next, enocytes and labial glands we will be learning with the help of next video. Thank you.